Good evening, ladies and gentlemen. My name's Peter Joyce. I'm Dean, University of Otago, Christchurch, and it's my pleasure and privilege to introduce each of these public lectures. We have these most years, except immediately post-earthquake. This year there are 6. This is the second of 6 public lectures. Tonight's speaker is Associate Professor Anthony Butler. Now, Anthony went through the Otago Medical School and did his clinical years of his medical degree in Christchurch, and I remember him from when he was a student, as an undergraduate medical student. He then trained in radiology through Canterbury District Health Board radiology services. But while he was doing his radiology training, he did a part-time PhD at University of Canterbury in engineering. And he and his father have been leaders in developing this Mars imaging. Perhaps the most notable recent achievement last year, the Ministry of Business, Innovation and Employment gave them a 12 million grant, which will be shared between University of Canterbury and University of Otago, to build the first human Mars scanner. I think I've already volunteered to be the first subject scanned in this new machine, and hopefully over the next few years we will have a prototype of a human model for this imaging. In very layperson terms, people say, what's it about? I said, well, in the old days, the X-rays were black and white and two-dimensional, and this is three-dimensional colour radiology as a simple description. But without more ado, hand over to Anthony, who will now talk to you about Mars.
Thank you, Peter, and thank you for coming on what's a beautiful Christchurch summer evening or autumn evening. So I'm going to be talking to you about Mars, which we consider a new form of medical imaging. I've got a picture here of a Mars image. That's a mouse. And it's got iodine and barium in its chest cavity. I'll explain a bit more about that as we go on. As Peter said, this is a collaboration between various universities. So University of Otago has been very key in the development of it, particularly around applications development, but has been getting more into the maths and the computing in recent years. University of Canterbury, with its engineering school and its physics department, have been very involved in the basic detector science behind this. And CERN, which I'll talk about during the talk, which is a particle physics group in Europe who discovered the Higgs boson. We've used some of their technology and we collaborate very closely with them. So to give an overview of my talk, I'm going to give you the goals of our team. I'm going to introduce the team. I'm going to explain what CT is in about 2 slides. And then I'll talk about the Mars spectral CT. And then I'll go through the possible areas of clinical impact. And I've actually just limited this down to places where we've done preclinical imaging in this medical school. So where I've got pictures to show you how it might actually work. Then I go off and talk about some physics and some engineering for a while as I go through the basics of the technology, and then I'll wrap it all up. So our goal is to provide new information about tissues. That means we want to measure things in tissues that other people haven't been able to. And as we do this, we're always thinking in the back of our mind, it's one thing to do it in the lab, but we want to do it in the hospital. We want to turn this technology into routine clinical practice. And I think that is a critical part of where being involved with a medical school changes your focus from if you were just involved with an engineering school. We've got to translate this to the clinic. We've got to try and get this into routine use. So to introduce the team, we're about 30 people across Christchurch. It depends a little bit how you count. We're currently 15 PhD students. I used to introduce Canterbury as the engineering and Otago doing the medical side of it. It's a little bit more blended than that now because while Canterbury is very largely physics and engineering, they've also got a biology department working with us. And Otago, which has done the cancer and the orthopedic research stuff with us, is, as I said, now getting a bit more into the maths and the computing behind it. Lincoln University is also involved, particularly as we get to moving from mice to larger animals. Unfortunately, despite Peter's request, we'll probably have to put a large animal in before we put a human in. And Auckland University, where they've got some detector physicists. We have a number of international partners. CERN, as I introduced earlier, which is a high-energy physics lab and one of the world leaders in particle detection. Mayo Clinic, which has some of our technology and has been publishing papers. Rensselaer Polytech, which is upstate New York and is they tell me, the oldest bioengineering centre in the US. Notre Dame and many others. We've got a number of commercial partners. One of the things that the government likes in New Zealand is they don't want to just do research for the sake of research, which it's a topic for another evening. They're very keen to make sure that the benefits get across society, and by that they mean they want to see companies formed, they want to see people getting jobs, and the universities have spun out a company to commercialise this technology. We work with a couple of local companies, Interlink Research, Shamrock, Fabtech, Assembly Specialists, which are local engineering firms. So a lot of the design happens within the universities, and then we get local industry to manufacture it, and not actually very occasionally, today's a nice example, we bring them into the university, we show them what we need done, and they go away and they do it. And last year, the University of Otago partnered with GE Healthcare to look at the clinical applications of this. There are now a number of major medical imaging vendors looking at where this fits on their 2025, 2030 roadmap. So I think that's actually very exciting because a few years ago people were saying that this would never take off. So to put a bit of background, this is about particle detection, in this case X-rays, and Ernest Rutherford started particle physics just across the road at the Arts Centre. So one of the nice things is when I go and visit universities overseas, if I'm in a physics department, I can say I've worked in the same place as Rutherford worked, and that usually gets me somewhere. Bates and Peters worked out in the electrical engineering and then more in through the hospital. This is actually before the med school was really, or as it was getting going. They were the first people to publish the use of Fourier transform in CT. Now that's actually quite an important step because through the late 70s it was a major method of doing the so-called CT reconstruction. And then later on it was replaced by a filtered back projection, which was a slight variant of it. And in 1972, unfortunately about a year after Hounsfield published his first pictures, they produced their first pictures. So those Christchurch scientists, in fact, got the idea, built it very independently, and I've even read the Nobel Prize citation for Hounsfield, and there's 2 paragraphs at the end saying, and we acknowledge that a group in Christchurch did it independently. So what is CT? CT is something you have in the hospital. We'll start back off standard X-ray systems. This is an X-ray system. I haven't seen one operating like this. There's pictures of it around. You have an X-ray source, which really haven't actually changed very much from this picture. They might change a lot in the next 20 or 30 years as technology changes. We have an object, and if we're in a hospital, we'll call that the patient. We have a detector, and in this particular case, it's a fluorescent screen. So the X-rays are hitting it, the fluorescent screen is glowing, and then we have a pattern recognition system, sometimes called a radiologist, who's looking at that and interpreting it. Okay. Now, what we've been focusing on is changing that detector system. Okay, so as I said, with that detector system, it glows. The more X-rays that hit it, the more it glows. As we got more advanced, we went to film, and that would record that black and white picture, and that meant the radiologist didn't have to sit in the X-ray beam. Things went digital, but essentially, the more it glowed, the more that was all you saw. This is a CT system. It's really just a 3D X-ray system. So you have an X-ray source here, shining an X-ray beam, and a row of detectors. This is what the one in the Christchurch Hospital looks like. I just pulled it off the web, and it's so-called GE multi-slice, 64 multi-slice. And that produces pictures like this. So this is a picture, 3D picture, and this is through someone's chest, and inside we can see that the aorta's showing up white, the bone's showing up white, tissues have shown up grey, lung and ear are showing up black. So that's a black and white 3D picture. So what happened in radiology? Well, CT got very, very good. Between 1998 and 2005, CT was growing at about 5 or 6% a year. They were just going in all over the place. And then it started slowing down. So what happened in that time? Why did CT do this huge rush of development and installed base? And I think we might have sort of about, have to count, the DHB's probably got between about 5 and 7 CTs in place scattered around like oncology in the community at the moment. So there's a big boost, but it stopped. And what it was what was called the slice wars. So CTs became very fast and they have very small voxels. So when I started as a radiology trainee in 2000, it used to take about 5 minutes to scan the head, and even longer to actually print it out on film, and we had slice thicknesses of 5 millimeters or 10 millimeters. Right now, if I walked into the hospital and I thought you had cancer, I could scan your head to your toes, probably in under a second. They're basically limited by the speed that you can accelerate the patient through the circle. You don't want them flying off the end of the bed. And you can do it in about 300, 400, 500 micron voxels. So as I said, they became fast and they became very high resolution. And that's a little bit like your digital camera. You used to care whether you were buying a 1 megapixel camera, a 2 megapixel camera, but once it got to 10 or 15 megapixels, no one cared, it was good enough. So anatomical or morphology or the shape is now really good. Going faster and getting more detail doesn't add much. We can take a picture of, as I said, the chest and the abdomen of a person under a second and get pictures like that, but it's still black and white. What people actually want to know is, what is the tissue? Is that cancer? Is that normal tissue? If it is cancer, is it growing or is it shrinking? What's its cell metabolism? Is it turning glucose over very quickly? Is it expressing cell surface antigens? These kind of questions. And then you move on to the treatment. I've given a cancer drug. Did it actually get into the tumour? I want to know what dose, what proportion of the dose I gave got inside the tumour. So researchers want to know the constituents of the tissue, the fat, the water, the calcium and the iron. They want to know about cancer and pathogen labels, and they want to measure physiological markers. It's not about shape, it's about biochemistry. So that means we need a fundamental change in the information provided by X-rays. Which leads me on to this. What is Mars spectral CT? So X-rays come in different colours, wavelengths, energies, frequencies, depends on your high school physics teacher. And in that respect, they're just like visible light. They're part of the electromagnetic spectrum. If we talk about visible light, everyone's happy to call them red, green, blue. But if we just shift to a slightly higher energy towards X-rays, we've got the same phenomenon, red, green, blue, different energies. And we can measure that. So this is a picture of a standard CT scanner. It might remind you a little bit of that very old picture I had of a 2D system. You have an X-ray source putting out a range of X-ray frequencies, colours or wavelengths. They pass through our patient, they're attenuated in the patient, and then you have a grayscale detector that gives you a single Hounsfield unit. This is the brightness in that voxel. Cutting edge technology, Christchurch Hospital put one in about a year ago, the so-called dual energy, and they have two X-ray sources putting out different colours that pass through the person, and you have two black and white detectors, and you get 2 points on the attenuation curve. This is actually a little bit like the way movies went to Technicolor in the 60s by having several different projectors instead of having it all in one pixel like your modern projector would. We're suggesting we move to the next step, where you go back to having an ordinary X-ray source putting out all the different frequencies or energies. They're attenuated, and then you have a very clever colour detector that can separate them out, and then you get multiple points on the attenuation curve. And that's really nice, because that attenuation curve is specific to every atom, which means you can work out what atoms are inside people. So as I said, spectral imaging is now possible. This is a fourth-year student we had a few years ago that's standing by a machine, and that particular machine was shipped off to the Mayo Clinic. We came up with an acronym, Medipixel Resolution System. Importantly, it's got energy resolution, spatial resolution, and temporal resolution. That is, we can measure the energy, we measure when the X-rays arrived, and we measure where they arrived. Current single energy scanners, as I said, are brightness only, that grayscale, and the spatial and temporal resolution. We called it Mars because it sounds futuristic and is easy to spell for the house surgeon on a busy night on call. So, some examples of this. This is that first mouse I showed you. We take some heavy atoms, and that means they're a long way up the periodic table, such as iodine, barium, and gadolinium. And these have very specific peaks in their attenuation called K-edges. Now, if we can measure that X-ray energy, we can use them in combination, so we can use two at once. We can start to use have new pharmaceuticals and say that pharmaceutical got to that part of the body. Okay? So, in this particular case, we put it into a mouse. This guy's called Mouse 12. He's famous in our group. And we put barium into the lungs, and we put iodine into the heart, and then we actually did a 2D X-ray, and you can see the barium in the lungs and the trachea, as well as the iodine in the heart and the big blood vessels, is all white. As I said, if you're only measuring brightness, they're just white. The bones are white because calcium attenuates a lot. So we can't tell which is which. If we do a 3D X-ray, a CT of that, we end up with a nice pretty picture of our mouse, but we still can't actually tell which is bone, which is barium, which is iodine, where they are. Is it going to let me move? Whoopsie. And here's the same mouse imaged on our scanner. We can see I've assigned colours to them, but in fact the red is where the iodine is, blue is where the barium is, white is where the calcium is, and where water is, we've made it grey. So that's 4 separate channels of information. That's not Photoshop, that's not someone going in and painting colours on after the fact. That is the X-ray energy that we've used to tell which material it is. So we've got quantitative material imaging. Now the next step is to take it through to humans. We can do a mouse. We actually want to do a human, which means we need to go from detector arrays this big to this big. This is going to take a wee while. It's not an easy task. We've promised the government we'll have it done by 2020, and I think it's probably going to take about 30 to 100 people in Christchurch to do this, which is an exciting challenge. It's quite nice. In essence, Canterbury University is doing a lot of the engineering. As I said, the medical school, we're doing a lot more of the mathematics and the computing. And then here within the health precinct, Otago University intends to host it to enable clinical trials. So we'll have it by 2020, and by 2025, hopefully we'll have done a few clinical trials, and I'm hoping I can offer Peter the first option. So what's the benefit of this? What is the clinical impact of being able to do this? What are we looking at the next 6 years? What are these first clinical trials going to be about? Well, we've been working with the orthopaedic department. So this is a picture of someone's knee replacement. They've had arthritis and they've had one part of the knee replaced. If we want to image that, let's keep our engineering easy. We'll do that in 5 minutes. Let's keep the spatial resolution quite low, do it down to 0.2 of a millimetre, 200 microns. Relatively easy imaging target. But what the orthopods want to know is, did this metal implant fuse with the bone? Now that's actually unfortunately really hard for them to measure at the moment, because you get all these artifacts, all these stars appearing off the end of the metal because of the way it interacts with X-rays. So in this case we took an implant that was actually developed by a local company called Osis. It was put in a sheep bone and then they grew together, and if I blow that up, you can see that we can clearly see at the sub-millimetre scale how bone and metal are fusing. And so right now, that enables a company like Osis to make their implants better. It allows the orthopods to start learning about what sort of shapes and how bone interacts with metal. And we can do that on the small scanner. And then when we've got a human scanner, we can start looking at these implants and how they go on people. So we could start doing a knee replacement in a person, follow them over time and say, this design of knee replacement worked better, this one didn't. Let's look at something else in the joints. This is some work, again with the orthopaedic department, on arthritis, in particular osteoarthritis. One of the problems with osteoarthritis is you actually don't see physical changes till many years after the biochemical changes occur. In particular, one of the proteins called glycosaminoglycan starts decreasing in the cartilage before you start getting wearing away of the cartilage, before the cartilage starts cracking. Now, if the orthopods knew in advance that that joint was getting unwell, they could actually start treating it. At the moment, they don't know until it's too late. So in this particular case, we've taken actually a bit of cartilage from someone who had their knee replaced in the operation, and we incubated it with a marker that shows up on our scanner, and we can see, and I'm going to make this picture bigger, where the biochemical changes are occurring. So you can, as I said, you can see here that the surface is actually pretty good. The other half of this knee was terrible, which is why it was taken out. But we can see that there's biochemical changes right there. And if this knee was still inside the patient, it'd be a good time to start treatment, because you can try and reverse the biochemical changes before you start getting the physical changes. We've done a bit of work with the rheumatologists on arthritis. So they have a number of diseases called gout, pseudo-gout, etc., that they have crystals forming around the joints. So we had a summer student a year or two ago comparing the crystals that form around here, and at least in a phantom, so that phantom means a bit of perspex, we put the crystals in, we can tell that they're all different. So we should be able to measure these. We then started working with at Notre Dame University, who have pharmaceuticals that measure bone turnover. And again, their pharmaceuticals are so-called nanoparticles with gold, hafnium, gadolinium, and we've been able to measure them on our scanner. So I'd really like to see that in first of all, some animal models and then later in humans, measuring bone turnover. And that would have a profound effect on people who are developing osteopenia and other bone disorders. We've been looking at the neck. We've been looking at heart disease and we've been looking at stroke. Okay, so heart disease or atherosclerosis is one of the world's killers. It causes stroke, it causes diseases in the leg, and it can cause a heart attack. In particular in the neck, if you get atherosclerosis, it causes a stroke, and people will come into the hospital with a mini stroke. They'll have a CT scan, and then you'll have to decide, do you do an operation? It'd be nice to know. So we've been looking at this bit of the body. Again, easy imaging, got 5 minutes to do it, people can lie still, down to 200 micron. What's going on inside that blood vessel wall? So some of these people have operations. They have this taken out, this bit of their blood vessel. It's quite a big operation, not without risk. And we've imaged it on our scanner, and we separated it out into water, calcium and lipid, and we compared it to staining, so I'll show you that. So we can measure how much fat, how much water, how much calcium is in that plaque. So if this was in the person's body, the question is, would that rupture, would that go on and cause a stroke? And we can now measure it in a way that you'd normally have to take out and do under a microscope, but we can do it inside the person. We started doing a similar sort of thing in that this is in fact with some sheep tissue from the butcher, where we wanted to measure the fat and the water content of the meat and separate them out. It's in fact exactly the same problem as doing it in someone's neck. So here we've got a 3D one. This particular scan was done in what I call the human energy range with the appropriate amount of filtration. So if this was a human neck, that's the quality of image we could get from their neck. We would be able to see that there's a big lipid-rich core here and therefore likely to rupture. There's a bit of calcium there that's stable. This is the direction to the heart, this goes to the head. So that, in 5 years' time, I'd like to be producing from a person. We've also started working with a group in Sydney. They produce pharmaceuticals which track to a single cell line called activated platelets. And they believe that these activated platelets are critical in which atheromas go on and cause a heart attack or a stroke. And so they produce they produce antibodies to these cell lines, and they attach gold to their antibody, and we can see the gold on our scanner. If I blow that up, this is an ordinary CT of that bit of tissue. It's from someone's neck. And one of these is calcium, and one of them is where the cell line is. If you have an ordinary CT, you can't tell which is calcium, which are these activated platelets. If you have a colour scanner, it's easy to tell. The one on the left has activated platelets, it's got microhemorrhage, it's a plaque at risk. The one on the right is calcium, it's unlikely to be a danger plaque. So what about the long-term promise? So we're working, as I said, with Notre Dame, and they've been looking heavily at cancer particles, where they will take surface binding antigens on their nanoparticles, inject it into the bloodstream, they'll go to a cancer, and on a black and white x-ray where they only got one thing to look at, they can say they can deliver their drug containing their cancer to the area. But also, all of their labels that they make, and they're funded to make a library of spectral pharmaceuticals, are different x-ray colours. So there's potential in the next few years to have multiple targets in the same time, looking for the different cell lines within a cancer. So that's where I hope we're going to go, or we were definitely going to go. I hope we get there quickly. So what about the underlying technology? Where did this all come from? So I'll come back to this slide, which is our happy fourth year student with his preclinical scanner, and show you a picture of it. This is what it looks like. There's one in the med school here and 3 out at Canterbury. They're a standard CT scanner. I showed you the picture of the big one. We have an X-ray tube here, an X-ray detector here, our colour camera. This whole bit rotates around, and we put it in a box. It looks a little bit like a barbecue. It happens to have lead around the outside, so you can stand beside it without having to wear any X-ray protection. This is an X-ray camera. It's called a Medipix camera, or contains a Medipix detector. It's what's known as a photon processing detector, or a photon counting detector, or a quantum detector. And as I said, high energy physics was part of this story. And the reason for that is to measure an X-ray beam. If you want to measure the energy, the best way to do it is to analyze every single photon one at a time. Okay? It's a pixelated detector, which is to say every pixel has its own electronics, and it's a spectral detector, which means it measures the energy of the photons. It's been developed by the Medipix 2 and Medipix 3 collaboration. New Zealand was a founding member of Medipix 3, and it will be a founding member of Medipix 4 probably later this year. And the goal of that collaboration is to transfer technology from high energy physics into medicine. Now, what intrigued or amazed even me was when we first met this group back in 2005, there was about 20 universities involved, no medical students, no radiologists, not even any medical physicists. They were basically all microelectronics engineers producing circuits, and they were doing a wonderful job. And we came along and said, well, let's actually turn this from the physics lab into an application and get it out there and get it into use. Came out of high energy physics, so I'm part of the so-called compact muon solenoid experiment. CERN itself is 70 countries, 6,000 scientists. They're famous for developing the World Wide Web, and a few years ago, 5,000 or 6,000 people got their name on a paper for discovering the Higgs boson. This is a compact muon solenoid. If I get my figures right, I think it's 1,200 tons. I mean, it's huge. It's 100 meters underground. This thing here in a clean room, these are my holiday photos, by the way. This in a clean room here is a multi-million-dollar detector for particle physics, okay? And in there is, in the so-called silicon barrel, are all these particle detectors that analyze the particles coming off from the interactions, one particle at the time, and that's the technology we borrowed and tried to turn into medicine. So here's going through that individually. This is one pixel. Our pixels are 110 by 110 micron. In fact, it's a combination of 4 of those. We can measure up to 8 energies simultaneously. There's 5,000 transistors per 110 micron pixel, which means it's got about as many transistors in it, in the whole chip, as we then combine it into a 120 by 128 array, which means that if we've got 5,000 transistors per pixel, a 128 by 128 array, we have about as many transistors as most computers, just in the X-ray detector. Now you go back to my first picture of the radiologist holding a screen. There were no transistors, it was all analog. The digital systems typically used in a hospital have 1 or 2 transistors per pixel, and we're taking it up to about 5,000. We can then tile these into an array. Our chips are 14 by 14 millimeters, and we can tile them into a linear array like that and make anything as big as we want. These are our current fingerboards, so these they're about the size of my thumb, and the active area will be the thumbnail. You can make a strip as long as you want, which means we can scale for 3 for mice and 100 for a human. This is a schematic of it. I will just quickly dive into some fun physics. What happens here is, this is the schematic. This is a sensor layer. A photon comes down, if you can imagine, hits in the top layer there and produces an electron hole cloud, and that's conducted down, if I've lost my pointer, into these electronics, and that cloud is analyzed, and that you measure when that cloud reached the electronics, you measure how big it was, and then you record it to memory. And it's doing that one photon at a time. It's pretty clever as well, because what happens is when that photon comes down, it doesn't interact typically in one pixel, it interacts in many. And it's split between, so one photon will go into two detector elements, like this, or in this case 4, that's where the photon's interacting. As I said, the charge is split across 4 pixels. one pixel takes most of it, and it decides, I'm the winner. And then all the circuits have to add that together and then allocate back to that one pixel. So for every photon that's hitting that detector, you're doing this addition and allocation. I'll come on to the speed that means to be done in a moment. So what does that mean? Well, if we take something like an americium source and you scan it with our detector, we can measure the X-ray spectrum very accurately. So we have a spectroscope. For those of you who did undergrad physics and did spectral measurements on things, we can do that in every single pixel. And as I said, we've got millions of pixels. If we didn't do that, all that complex adding of charge together, the spectrum you measure is blurred out, which is that dotted line. So really having these fast, intelligent electronics make this sort of technology possible, which means you've got new clinical applications. And it goes fast. This is one of our partner institutes. Thomas has been to New Zealand 2 or 3 times to work with us, and this is some work he did in X-ray laser in Germany. We have to do that at about about 1 million photons per second per square millimetre. So all of that adding of charges together, allocating to a pixel, you're doing millions and millions of times per square millimetre, and you're doing it for the whole scan, which means Medipix 3 is really, really fast. And that gets me back to where we basically began. We can do it in animals. We want to do it in humans. I think we've got to the point, and I guess the fact the government gave us as much money to do it, we've convinced our funders and our partners that it's actually worth doing. There are going to be health benefits, that people are going to lead better lives because of this. People will live longer. But actually, for me as a scientist, I think this is a very fun and very challenging project. So in conclusion, spectral CT is the future of X-ray imaging. Black and white imaging, I think, will just go in the next 10 or 15 years. There's no penalty in measuring the colour. Once the technology is there, you can do it. For example, when you had black and white, when Kodak brought out colour film, everyone gave up on black and white film. Preclinical work is certainly clarifying the benefits, and I think that's actually kind of an interesting one. One of my colleagues, Nigel Anderson, jokes he's got a list of 30 to 50 people wanting to use the machine, and he's not sure which ones are going to work, so he has to work out, this is a likely good candidate, we'll try that, and then a few of them do work. And New Zealand's building a human scanner, so Peter here is retiring in a few years, and sometime early in his retirement, I hope I can stick him in a machine. Thank you.
Thank you very much for that, Anthony. He's left good time for questions and discussion. So if people have any, for those who can follow the physics and anything else or the applications, are open for discussion.
Louise?
Just one question. How do you anticipate the doses are going to relate to the doses that we're seeing in CT scanning?
There's lots of ways to answer that. The short answer is going to drop dramatically. The reasons for that is probably actually what... Louise is a radiography tech who works here at the hospital. So I've worked with her before in a different context. The reason for that, we can stop the X-rays just as well as any other detector. So we're as efficient. But on top of that, we go to smaller size pixels, and that means you get less noise through the image. You cut out what they call the mid-frequency noise. And then on top of that, you've got energy information, so your soft tissue contrast goes up. So that really, really helps. You get much better signal to noise. We can measure down to the so-called Parsonian limit, so you can see the quantum model in the X-ray beam. Okay, and that means that your image processing, your mathematics changes because you're now working at the noise limit. And so you can do fancy tricks like undersampling your data. And so one of the groups we've worked with a little bit, which at Stanford, they believe the dose will drop by 90%. My personal view is that that's probably achievable, but the bigger benefit is we're actually going to be able to do more with it. So when I talked about those slice wars, the X-ray dose of the population went up as we did more scans, and the scans had a higher X-ray dose. But the benefit to the population was massive. We could get rid of a whole lot of intervention, so catheters and needles that were being put into people, and do it on an X-ray machine. So even though you had a higher dose, the clinical benefit was much, much better. In this case, I think we're actually going to win in both ways. I think the X-ray dose will drop dramatically, and I think the amount of information we provide will go up dramatically.
Yeah?
How freely available is, or what is freely available to me from what you've been talking about?
I'm not sure I understand what you mean by freely available. We publish a lot of our work, right? So you can read about it and things. But as you're saying, as someone who wants to come and use our preclinical scanner, what's available? Or okay, so the
Especially for me as a non-insured, in other words, what's freely available within the public health system to make my health status better.
Okay, so this is still at a research level. By 2020, we want to be conducting clinical trials. I keep joking with people in the DHB that we need to have one in clinical production by 2025, but that's a different question. So as technologies go through, they become available to the public sort of late down the process. So the first human scanners will be used for research because you actually need to understand how does it fit into the process of disease management. And I guess, you know, one of my personal goals here, when I first got involved with this sort of technology, I was still a radiology registrar. I was still working on Friday nights reporting car crashes, and I could see this technology and thought, how on earth do you take it from a physics lab through to clinical use and shortening that time down as much as possible? And for a pharmaceutical, that's, I think, about probably 15, 20 years typically. For technologies like this, if we can get it into routine clinical practice by 2025, we're doing pretty well.
This will help your children and grandchildren. It may not help you.
Yes. Yeah, single source scanner, yeah. And in fact, one of the nice things is that we actually can use a very cheap source because we want the equivalent of white X-rays. Whereas the X-ray sources that are being used, say in a GE dual energy system, are actually quite expensive because they're trying to filter it down to be as monochromatic as possible. Whereas we actually want the broad range of energies. Our current tubes use tungsten. Yeah, just standard off-the-shelf tungsten tubes. And in fact, you know, we thought about how we're going to build a human one. One of our engineers worked out at Lincoln University, and they've got a CT scanner for their animal work, and he basically took an old GE one and got it working. And you could have essentially do the same. I don't think we'd want to do it that way, but it's a possible way to do it. Standard source. Nothing ever becomes redundant in radiology. One of the things about radiology is it grows remarkably quickly. As I said, I started in the radiology department in 2000. I think the department must be 2 or 3 times as big because it learns new applications as a specialty. So nothing really gets replaced. What happens is you find new places to use it. The most common study in our radiology department still is a chest X-ray because you can do them cheaply, quickly, you can learn a lot of information about them. So in that respect, I don't see anything being replaced. What this allows you to do is get some of the information that you got off an MR and some of the information you got off a PET, but a whole lot of information you couldn't get any other way. So what I would think is it's going to reduce things like biopsies, and we're not sure what's going on, so we'll wait in 2 weeks to do another study. So you've got another test you can do and make that diagnosis earlier and start treatment earlier. Mm. And you've got a pixel size of 100 microns. Mm. How will it be down to 10? I'll start with an anecdote. When I actually did an undergrad physics degree as well, and I had summer studentships from physics into the medical school ones. I actually worked in oncology, and I had a spectroscope, and I used to have it in my talk. The picture of the spectroscope was this big for a cool germanium thing, a trolley with the electronics on and a computer beside it. And now that's, as you say, 110 by 110. One of the questions that then comes up is, why would you go smaller? As I said, the charge cloud in that sense is quite big. So you're going to have to add across more areas, across more pixels. But I've got a colleague from Prague who says charge sharing is my friend because that's how I analyze the particle. So you may go down to, well, he's pushing for Medipix 4 and Medipix 5 to think about having pixel sizes of around 10 to 15 micron. The electronics is possible, the bonding onto the sensor layer, the interconnects becomes pretty challenging. And then you actually have to think of not a simple scheme that I had of just looking at the nearest neighbours, but actually passing the charge across a very large number of pixels. So we will go that way. The question is when and quite how, and yeah, but there are certainly discussions about how to do it. Yeah. I think within 15, 20 years, there'll be no such thing as a black and white CT. Okay, CT is rapidly becoming the mainstay for trauma imaging anyway, particularly anyone who comes in, I forget exactly what the DHB protocol is, anyone with 2 body parts injured or mechanism above a certain amount automatically gets a CT, because otherwise they used to come to radiology and then they go back to ICU and then back to radiology. We just CT them up front. I think we're likely to use a system like this in the future, because there's no point in doing a black and white image when you can do a colour image. As I said, it's if you've got black and white film and colour film, well, you'd always use a colour. Okay, the only reason you potentially wouldn't early on in the technology's life is cost. But in 20 years, this technology will be the standard mature technology. It'll cost the same as everything else, so you'll do it. So yes, it's going to be used in trauma.
All the grant applications that you and your group put in cross my desk for signing off, so I see what projects are coming up. But one of the ones that struck me recently, because you briefly talked about different cancers, you know, one of the issues is how quickly you can see a cancer shrinking or not shrinking, and also related to that, the extent to which drugs are only hitting the surface of the cancer versus actually getting into all the cells in the middle. Do you want to elaborate on that?
Yeah, I will actually. I didn't include cancer except for one sort of diagram, because until a few days ago I didn't have a pretty picture. And that's essentially when you're, of course, preparing a presentation, you want pretty pictures. So one of the things about cancer in general is you want to know whether your drug got in, and you want to know the vascularity, what the blood vessels are like in the tumour. So we've been working with a bunch of groups here, the gynecological cancer group led by Pete Sykes. There's a group, Marguerite and Gabby, looking at lung cancers and melanomas and things. And they produce nanoparticles, or some of the groups do, where within this particle, let's say 15 nanometers, you can pack in gold and then you can pack in drug. Okay, now we can see the gold quite happily, so if you inject that into the person, you can see what the uptake in the tumour was, and that gives you an idea of how much of your drug actually got there. Okay, now there's also the ability, which is what Pete Sykes is doing, you might have signed this off a week ago, where he's producing nanoparticles, so those envelopes, but on the outside of them he's putting luteinizing hormones, so they'll selectively uptake within some of the gynecological tumours. So the gold gets in there, and we can see that those cell lines exist in the tumour. Okay, so that allows you to say that's that type of tumour. And I was talking earlier about, you know, maybe getting rid of the need for biopsy. That's cell tracking. That's saying this type of cell line is in this tumour and this part of the body, and therefore we need to treat it with this kind of drug. So it's still at the early stages, but it's very much on the minds of the researchers here, because they're actually trying to do this right now. And the picture that I don't quite have, because the student showed it to me on Monday this week, is a lung cancer in a mouse where the gold has gone to the outside of the tumour, and we can actually start measuring the vascularity of the tumour. What are the blood vessels like? And he's very, very excited because he's been trying to do that for a while. And I'm quite excited because I've been wanting him to do that for a while. So it'll be interesting to see where that work progresses over the next year or two. Because we need to have this preclinical work done in time for a human scanner. We've got to keep looking at all these applications, so when we've got a human machine, we know what diseases we should be studying on it.
The question down here.
Yeah.
Do you think you'll be making any other projects?
This is my son. I think that's for the audience and for him, that's a bigger question perhaps for the university, but the university has grown what it calls bioengineering and nanomedicine a lot in the last 5 years, and it's a new direction that medical research is going. There's a lot more looking at how computers help medicine, how mathematics helps medicine, how various bits of technology such as implantable devices, tissue engineering, etc. can help. And that's very different from the sorts of research were done when I was an undergrad at the medical school in the 90s. So, yes, Dylan, I think there will be other projects. I'm not sure when and what.
Other questions, comments? Well, if they're not, I'd like to thank Anthony in the normal way, but certainly from where I see this as one of the most exciting developments where Christchurch may again lead the world in this area, and I think it's a very interesting example of collaborations across Canterbury University, ourselves, others, and industry in terms of the requirements for the government funding. So thank you, and Phil Butler down the front from University of Canterbury for all the work that's been done over the last 10 years really. But thank you to Anthony.